Hi, my name is Natasha Chang, and I'm very happy to be able to give you a research update on our Defeat Duchenne Canada funded research project entitled Inducing Stress Granule Formation in Muscle Stem Cells to Treat Duchenne Muscular Dystrophy. The research in my lab is really focused on understanding the biology of the muscle stem cell. And this cell is also called the satellite cell, and it's pictured here. And this is a very rare population and a very specialized cell population that is responsible for regenerating the muscle in the context of exercise or injury. And so in the lab, we are very interested in understanding the molecular mechanisms that regulate muscle stem cell function and then to expand on this knowledge and understand how the muscle stem cells become dysregulated in the context of disease. And one of the diseases that our research focuses on is Duchenne muscular dystrophy. So DMD is a lethal genetic disease that's characterized by progressive muscle wasting, and it's caused by mutations in the DMD gene that result in an inability to generate a protein called dystrophin. And what we know is that dystrophin expression in the muscle is very important for membrane stability and the strength of our muscles. And without dystrophin, the muscles are weaker and more susceptible to contraction-induced damage. And what our lab has shown, and what other labs also have demonstrated, is that the muscle stem cells in Duchenne muscular dystrophy are also impacted by the loss of dystrophin. And this results in an inability of the stem cell to fully commit to the regeneration process and to make muscle. And so one of the experiments that we did in my lab is to understand how are stem cells affected by the loss of dystrophin. And what we did was an experiment that's called single cell RNA sequencing, where we're looking at the individual gene expression of individual muscle stem cells. And we did this in the context of Duchenne muscular dystrophy and compared this to healthy muscle stem cells. And what I'm showing you is the result of this experiment where each dot that you see here represents an individual cell. And so the main conclusions that we got from this work was that the gene expression signature in Duchenne muscular dystrophy muscle stem cells is significantly different than the gene signature in normal healthy muscle stem cells. And what it told us was that these DMD muscle stem cells have an impaired ability to undergo myogenic differentiation. So they're impaired in their ability to make muscle. And so in the lab, the way we are approaching DMD is to understand how the stem cells are dysfunctional, and then to devise strategies to improve the differentiation capacity of these muscle stem cells. And by doing so, what we're hoping to do is restore their function and help them to make better and stronger muscles in the context of DMD. And what we did for this specific project is to take work that's already been done in cachexia and mouse models of cachexia, where they used a specific drug that was known to induce stress granule formation. And they found that this drug was able to improve differentiation of muscle cells in cachexia. And we asked whether we can repurpose this drug and use it in the context of Duchenne muscular dystrophy. And so one of the initial experiments that we performed was just to ask if we use this drug on DMD muscle stem cells, can we improve their differentiation? And what we found was that, yes, we were able to improve their differentiation with treatment following adding the drug. And this was done on cells in a dish. And we're here calculating the number of fused nuclei as a measurement of their differentiation capacity. We then took this one step further and performed a specialized muscle stem cell experiment that we have available to us in our lab, where we're then culture the muscle stem cell along with their associated muscle fiber. And we can observe the fate of these muscle stem cells throughout the culture time period. And so what we saw was that after 24 hours in culture, we were seeing an increase in the number of committed muscle stem cells after treatment with the drug. And by 48 hours, we were seeing this shift from commitment towards differentiation, indicating that in the presence of the drug, these muscle stem cells were now becoming more differentiated. And finally, we wanted to go from in vitro models to in vivo models. And so here I'm showing you experiments where we've treated DMD mouse models with the drug. And what we were able to see was that following treatment with the drug, we actually saw an increase in muscle weight, which indicated enhanced muscle regeneration. So, what we've really been focused on in the past year was really to understand the molecular mechanism of how this drug is working. And what we know about stress granules is that they impact the ability of our cells to make protein. And so what I'm showing you here is called a polysome profile, where we're looking at the status of protein translation within the cell. And we are comparing the polysome profiles of our control cells with our drug-treated cells. And you can see that they have vastly different protein translation profiles. And so what we are now doing is to take these profiles and then understand which proteins are being translated differently in after treatment with the drug. And so we're doing this by performing quantitative PCR experiments as well as sequencing experiments. And this will allow us to know which proteins are affected when we treat with the drug and how this leads to improved differentiation. So I'd like to thank my research team for all their efforts in supporting this project. In particular, this project has been led by a PhD student in my lab. Her name is Romina Filippelli, and she had the opportunity this past year, to attend the family forum that was organized by Defeat Duchenne Canada. I would also like to thank our collaborators for their contributions toward this work, and to everyone in the Defeat Duchenne Canada community for really supporting our research. Thank you very much.